So it's 7:02. I think we should go ahead and get started. So welcome to Engineering on Tap Zoom edition. I'm Leanne Hartman, and I oversee the college's alumni relations programs for communications, for events like Engineering on Tap, volunteerism, and giving. And I'm so glad you're here with us tonight. We launched this series last year with the idea that we can gather casually to enjoy a drink and learn something new about the College of Engineering. Of course, COVID-19 had other plans, but we are proud with the way that we've been able to pivot and adapt and connect with our community in all places over the world. We do have a few housekeeping notes, as you will see. We'd love to know who you are and where you are joining us from this evening. So please drop in your degree and your graduation year, or if you are a faculty or staff member that didn't graduate from UC Davis, maybe write that in the chat as well. Of course, the great thing about virtual events is that you can attend from the comfort of your own home and with your drink of choice. Tonight I have water and a coffee cup. But please use the Q&A button to submit your questions. It's a little bit challenging to ask questions in this environment, but we want to make sure your questions are answered. So please use that Q&A button below. And then after each presentation, we will answer your questions. And then at the very end, we'll all come back and you can raise your hand, use that raise your hand button to ask a live question, or continue to use the question and answer button. Before we get started, I thought you might be interested, although we have 41 or so participants on the call tonight, we actually have nearly 30,000 alumni from the College of Engineering located worldwide. Over 70% live in California, with 41% living in the Bay Area and 19% living in the greater Sacramento area. So you are in good company if you are interested in networking and connecting with fellow alumni. Please make sure you are following us on social media, you are connecting with alums on LinkedIn, and you are staying connected with us for other future events like this, as well as volunteer opportunities. So let's get started. I am pleased to have our esteemed faculty joining us tonight. They represent 3 College of Engineering departments: chemical engineering, biological and agricultural engineering, as well as electrical and computer engineering. Before we started this presentation, I had a chance to ask how long everyone has been with the university. And believe it or not, Dr. McDonald holds the record with 35 years. And Dr. Priya Shah has joined us most recently with only 3 years under her belt, but collectively they have spent 97 years with the College of Engineering at UC Davis. So that is quite the brain trust. And I am so thankful that they are spending some of their precious time here tonight to talk about how they have taken their research that they normally do and have pivoted to help solve some of the greatest challenges that our society is facing that's been brought on by COVID-19. I know that I am extremely proud to be among so many problem solvers and change makers, and I'd love to dive in to the presentations and let you know how our faculty are changing the world in real time. So with that, I'd like to turn it over to Dr. Shah to get us started.
Okay, well, thank you, first of all, very much for the introduction and for the invitation to share some of our work with you today. So what I'm going to be talking to you about, along with my collaborators, Professor Karen McDonald and Professor Roland Faller, is our work. If you could advance the slide, I don't know if I have control over it. is our work on producing proteins for COVID-19 therapies and testing. And really, can we improve this process? And so we're using each of our particular skills, my group's experience in producing really difficult to work with viral proteins in particular, Professor McDonald's group's experience in producing proteins in plant systems that are really easy to manufacture and scale up. And then also Professor Faller's group's experience in using computer simulations to understand how these proteins structure and function affect quality. And so with that, I do want to tell you what types of proteins we're producing. If you could advance the slide, please. And so the types of proteins that we're really interested in producing are called glycoproteins. And glycoproteins are literally proteins that have a sugar molecule or a glycan covalently attached to them. And so here, this spaghetti cartoon that you see here is a glycoprotein called ACE2 that we'll talk about in a couple slides. But the colored parts that are gray, orange, and purple or light blue, those are the protein, whereas the neon green parts are the sugar molecules that are covalently attached, or these glycans that give glycoproteins their names. And so these proteins are important for fighting COVID-19 because they make up a large number of our therapeutic strategies and our testing strategies. So for example, recombinant antibody cocktails, like the ones that Regeneron and Eli Lilly are making, those are glycoproteins, as are interferons that we use to boost the immune response early in COVID-19 infection or disease. And then also ACE2 based receptor traps are another example of a glycoprotein therapy strategy for COVID-19. And I'll talk to you more about what a receptor trap is. And then finally, in terms of vaccine, these viral proteins are also glycoproteins that have to be produced for a vaccine and also for serology testing, which is where we're going to focus. And so there are two proteins, if you can go to the next slide, that we're really interested in producing. The first is called spike, and spike is a protein in SARS-CoV-2. And so this purple cartoon here is a picture of the SARS-CoV-2 virus particle, and decorating the outside of it are the spike proteins. So this is a glycoprotein that allows the virus to attach to a human cell and infect it. And so here on the right-hand side of the slide, you can see a close-up view of one of those spike proteins. And this entire protein is important for the structure of the virus and getting into the cell, but it's also a major target for immune recognition. And so when an individual is infected with SARS-CoV-2, that individual is going to start producing antibodies against this glycoprotein spike. And the antibodies are going to bind all over this spike protein. And so being able to produce this large glycoprotein is going to be really important for serology tests that will actually detect these antibodies that humans produce if they're immune to SARS-CoV-2. And so this is a really difficult viral protein to express. And so one of the things that we're really interested in figuring out is whether we can come up with strategies to improve the production of this protein. If you can go to the next slide. The other protein that we're really interested in producing is a modified version of ACE2. So ACE2 is this human protein that's in this cartoon, it's this orange protein that's expressed on the surface of human cells. And it is the protein that SARS-CoV-2 binds to. So spike, which is the red protein here, it binds to ACE2, and that is what allows the virus to infect a cell. And so one of the exciting therapeutic strategies that people are talking about right now is the idea of making decoy ACE2 viral traps. And so the idea here is that we would engineer an ACE2 dimer, so two ACE2 proteins connected together covalently, and inject these into patients that are infected with SARS-CoV-2, very much like a recombinant antibody cocktail. And so these proteins would bind to the SARS-CoV-2 viral particles and take them out of circulation, effectively trapping them. one of the reasons this strategy is more exciting than the recombinant antibodies that are currently being tested and being authorized for emergency use is because of the way ACE2 works. So if the virus evolves or mutates so that it no longer binds ACE2 and avoids the therapeutic ACE2, it's also no longer going to be able to bind the ACE2 on human cells itself. And so the ability of this virus to actually infect human cells would also be reduced. And so this is thought of as maybe a potentially more robust strategy for therapy. And so this is one glycoprotein that we've already produced in the plant system, in Professor McDonald's group, in a mammalian system in my group, and we've also modeled it in Professor Faller's group to understand its function. And so with that, I'm going to hand the stage over to Professor Karen McDonald, who's going to tell you a little bit about the experimental systems that we're using to produce these two glycoproteins.
Thank you, Priya. Could I have the next slide, please? Thanks. So good evening, everyone. It's a pleasure to be here. And yeah, I have the distinction of being the oldest person and the only one wearing their safety glasses in the photos you saw earlier. Could you go back one slide, please? Thanks. So what I'll talk about is how we make these proteins, these glycoproteins, in two different systems. Professor Shah's group has a lot of expertise in using mammalian cell cultures for recombinant protein production. And one of the ways that they do this is transiently. So the idea here is, if you look at the little cartoons there, once you know the amino acid sequence for the protein you want to make, you can sort of reverse engineer it and determine what the DNA sequence is that will code for that protein. And so that is shown as the little blue part of the little blue part of this circle here, which is a piece of DNA. And that's added to mammalian cells, typically Chinese hamster ovary cells are used, along with transfection reagents that allow that piece of DNA to get into the host cells. They make the recombinant protein, it's secreted into the culture media, and then through centrifugation, you can harvest the supernatant and then purify it using liquid chromatography methods. So some of the advantages of this method is, one, speed. The batch culture time is about 7 to 14 days. So you can design a protein quickly and make it relatively quickly as well. Another advantage is scalability, because once you have a genetic system that you know works and makes the protein you want to make, you can then create stable transgenic mammalian cells. And these are well established. There are large scale production facilities. For example, Genentech in Vacaville has one of the largest mammalian cell culture facilities in the world. And you can use those to make very large quantities of antibodies or ACE2 FC receptors, whatever you're interested in. Another advantage is the high productivity. These cell lines, the stable transgenic lines, can produce grams per liter of the protein. And also protein quality. Professor Shah mentioned the importance of the sugars that are added, and mammalian cells can add those sugars in the same way that human cells do. Lastly, flexibility. Transient transfection could be used to produce many different proteins on a short time frame. And this is important because we're looking at variations of the spike protein to see which ones are expressed more highly and which ones would be more effective. Next slide, please. So in our lab, we use transient plant-based production. I know there are some people in the audience who have a lot of experience with this, some of my former students. But the idea here is that you can grow plants. These would be, for example, what's shown here is a relative of tobacco, Nicotiana benthamiana. And you can use a soil bacteria called Agrobacterium tumefaciens to transfer the genetic instructions to the plant. And then through the process of agroinfiltration, which I'll describe in a second, you can get those agrobacteria to transfer the DNA to the plant cells. You can incubate those plants. And then after the protein is made, you can extract and harvest the protein. Here again, speed. You can make these proteins within about 9 days. Scalability, large-scale facilities are actually in operation. And cost, we've done some techno-economic modeling that shows that you can reduce the cost of both production and the capital investment required. And finally, flexibility, because you can use different agrobacteria, you can mix and match bacteria, and you can very quickly test new proteins or antigens. Next slide, please. So if you could click on the, this is a video, so if you could click on the video to start the video, that would be great. So this is just a lab scale version of how the agroinfiltration process works. The plant is submersed in the agrobacterial solution and a moderate vacuum is applied. That allows the air that's in the interstitial spaces of the leaf to leave the plant leaf. And then when that vacuum is released, the agrobacterial solution basically perfuses throughout the leaf tissue. You can then take the plant tissue out and incubate it, and it's producing the recombinant protein during that 4 to 7 day period. And then once the protein's produced, you can extract and purify it. Okay, so now I'll turn it over to Professor Roland Fowler, who will talk about how the molecular simulations help in this process.
Good evening. I know it's called engineering on tap, but I don't have a tap, so sorry, I only have a beer in the can here. Next slide, please. So my group does computer simulations. And the big advantage computer simulations actually have is that we really know what the system is doing on the local scale. We really know everything about our system. However, this is on scales which take forever to actually do the simulations. So the first thing we have to do is we have to select the right model. And as we are talking here about proteins and glycoproteins, we really have to have every single atom of the simulation represented. And although our computers are really fast and we use a supercomputer here at UC Davis, HPC1, and we are getting a new one quite soon, and we also use computer simulations at national labs, we still need experimental structures in order to be able to do the simulations. We cannot just put the atoms into the system and hope that everything works. So we need to combine the models, and then we are using Newton's equations, and we use them billions of times. So the computer does calculate where all the atoms are and how they are moving a few billions of times. And this sounds a lot, but actually it isn't. A few billions of times only means that you can simulate at best these days something like 1 microsecond. 1 millionth of a second is very long on the time scales we are living on. So therefore, we need to make sure that we have the right model, that we actually can do these simulations. But then we can analyze the data and directly compare them to experiments. So we can directly compare what the experiments are doing. We can directly compare that to the simulations. And the advantage of the simulation now is we can change things which are much harder to change in experiments. We can really control things. We can, as we talked about earlier, we are interested what these sugars are doing, which are connected to the protein. And if we use different plants or different mammalian systems for the same protein, they will have different sugars. And in the computer simulation, we can just take one sugar off and put another sugar there, which experimentally is not possible. And then we can predict what actually is happening if we change the system very, very local. Can you please go to the next slide? This is also a video. This is actually a video of such a computer simulation. And this is just the protein. This is actually a piece of ACE2, this receptor, which you saw earlier. And we don't show any water. So in reality, this is in water. And we are talking here about a few 10,000 atoms. And this is a few nanoseconds, so a few billionths of a second to do these simulations. And it takes still about a week on a supercomputer to do this simulation. So, but then you really see, we individually see which part of the protein is doing. And then we can see how this actually binds to spike. And if we do a slight change on the protein, how do the interactions with spike change? Can we go to the next slide, please? And then we can compare to experiments. Again, on the left-hand side is a piece, in this case, of the spike protein, which is relevant for serology testing. And on the right-hand side, there are some experimental data. And we can directly compare the experiments and the simulation. And then we analyze them together so that 1 + 1 is much more than 2. The experiment and the simulation alone are already very, very powerful. But together, they are way more powerful than each one by itself. And so that is really the reason why we are working in this big team. And we are meeting once a week with all of our students, virtually, of course, again, on a Zoom call, and then go around and see what everybody has been doing and what ideas we have. Can we go to the next slide? And yes, these are actually the people who are involved or who have been involved over the last few months. And we actually got some money from the UC Davis Office of Research for what is called a craft project to actually get this work going. And now we are happy to take our questions. Leanne?
Yeah, we thank you, professors. I guess people are being a little bit shy today. I actually had a question. So you mentioned having a bit of an investment from the Office of Research, and I would be curious to know what additional investments would you need to be able to accelerate your research, or get it out of the lab and into industry and into the market, and sort of what kind of time horizon would that look like?
Karen, do you want to answer that, since you've got something that's probably the closest to getting out there?
Well, I would say that it depends. If you're talking about the ACE2FC, which is a human therapeutic, it requires a lot of money, a lot of resources, funding to move from research and development into maybe animal trials, and then finally into human trials. So that kind of project really requires a significant investment upfront for just the research and development, but then also partnering. You really have to find a company to partner with to move it into clinical trials. And yeah, it's an expensive, and usually it's a very long process. With SARS-CoV-2 and COVID-19, it's been amazing how fast both vaccines and therapeutics are moving towards actual use in humans. So it's great.
Great. Thank you. We are getting a few questions. First question, how well does the ACE protein replicator spread in a test environment? What volume of spread in the body does it need to achieve to effectively prevent infection?
Do you want me to answer that, Priya? I can probably take a shot at it. So this is exactly why research would need to be done. We really don't know. We know that it binds, the ACE2FC binds to the receptor binding domain of spike, but we don't really know how it would be distributed in the human body and how quickly it could find virions in the human body. So that's where a lot of research would still need to be done.
Okay. And then Michael has a question. Is there any way to simulate possible side effects of new proteins with simulations? I understand that would be brutal without existing libraries, but I don't know if those exist.
Essentially, the problem would be that we would need to have a model of all potential other proteins in the human body with which our protein would come into contact. In that And as that is very unrealistic, I don't think we are there yet.
I would like to add that I think there are some interesting computational approaches to predict side effects that are not from a molecular dynamics perspective. So they're more machine learning type computer experiments that mine the existing literature for potential side effects or potential connections between different drugs and different genes, for example, that have been used to actually predict adverse events and drug interactions by other groups. So there are computational approaches, but I think at the molecular level, yeah, that would be very challenging.
Okay, last question before we move on. It's for everyone. How much do government or company regulators require both physical testing and modeling simulations for potential treatments to receive funding?
Yeah, that's a great question. I think that with the physical testing, that is essential. Regulators aren't going to allow anything to be used, even in human trials, until they've been tested in animals. So the physical testing is really critical. I don't know how much the molecular dynamics simulations are used in the regulatory process. I don't know, Roland, if you have more of an input on that.
No, not yet, really. In this case, in other areas of engineering, this is done much more often yet. But as of course, human health is something that we have to be very, very careful. People don't use molecular simulations yet directly in the regulatory side. It's really kind of on the exploratory early research.
Great. Thank you all very much for sharing your research. Before we move on to Professor Sun, I want to give a quick shout out to our attendees in Phoenix, Arizona, San Diego, California, and Salem, Oregon. I've been stalking the chats and I want to welcome you to our event. Thank you for tuning in. I'll have a couple of shout outs later. Professor Sun, are you ready?
Good afternoon, everyone. Thanks for the opportunity to speak to you. And also my research related to COVID-19 is on the clothing materials. And in fact, it's more specifically for the face masks. I'm going to introduce the work on those so-called daylight-induced antiviral and antibacterial materials. Next slide. It doesn't work from my side for some reason. Yeah, and this is just a brief summary of my research. And actually it's focusing on the personal protection, mostly for professionals. And we work on those two sides. One is for the protective clothing materials. Second one is for sensors that can provide you the alert. And also the sensors that would have the detoxifying functions as well, that you will be able to see, and also you will be able to have the protection and simultaneously. Next slide, please. So we're going to talk on these face masks materials. And as we all know now, and face masks are considered as a PPE, and where everyone is using cloth ones and also the surgical and even N95s. Regardless which one you are using, but all of them are we call the filtering materials, serve as barriers to block those virus or the microorganisms. But all these live virus and will still keep alive on surface of the materials, which brings in another risk of infection to the users if you reuse it or improperly touch and also dispose it. And the idea here is to create what we call the biocidal surfaces. I will also introduce what's called a biocidal. And more specifically, we're introducing these functions by using so-called daylight induced, because we use light as a driving force to produce some of this real effective agent to kill the virus. So first is the biocidal materials, as I'm showing here, is that actually can kill the broad spectrum of the microorganisms. And mostly this would the functions equivalent to our disinfectant. And if you know that the disinfectants we use at home, we have the chlorine bleach, we also have the oxygen bleach. But we can't use those directly. In fact, we have used those, but I want to tell you that we are using the green and oxygen bleach indirectly. How do we do is we use some chemicals and let the chemical to produce oxygen bleach. More specifically, we define them as called reactive oxygen species, including hydroxyl radicals, singlet oxygen, and hydrogen peroxide. And those chemicals we use on the surface of the materials are safe, and even some of them are edible. Next slide. And so the first one actually I want to mention is the cotton. And the cotton, this is the real fabric we made is in the color. And also we put this, our symbolic structure of the UC Davis. So this face mask materials have those features: daylight induced biocidal functions, and also washable, reusable, safe to use, and also non-harmful to the people. So that's just recently published. Actually, ACS put a news release last week, and for this specific material. Next one. So you're going to see here is very simple chemistry, and we use cotton fabrics here is because most of the public are using the reusables, and we all wear this. And we put some kind of a cationic species, and those cationic species are the one similar to the softeners we're using, and as in the fabric in the laundry. We put those charged species on the fibers and brings in the cations and with positive charge. And those cations will strongly interact with one of the chemicals, the pink, red color is called the raw spingle, which is a biological chemical widely used in the medical treatment as well. This raw spingle is a so-called photosensitizer, and under daylight, it could produce reactive oxygen species. More specifically would be a singlet oxygen. Next slide. The singlet oxygen is very lethal to microorganisms. I'm going to skip this photochemistry, but just use the cartoon to show you what happens on the surface of the fibers. And it carries a positive charge, and the positive charge obviously attract those red dots, which are the raw spingle. Interacting with the oxygen, which are triplet structures, it will generate a singlet. This is on the daylight. The singlet oxygen will kill, and effectively. More interestingly enough, that the positive charge shows attraction to the microorganisms. And here shows you the two figures. One is with the increased amount of the raw spingle put on the fabric, you're going to see the high amount of this reactive oxygen species, the singlet oxygen generated. But with more of this raw spingle, the red dots onto it, you're going to shield this positive charge, actually will reduce the attraction to the microorganisms. So we don't want to put too much. As you see here is the amount. Once you exceed a certain amount, your interaction of this surface with the microorganisms will reduce. So mostly we select a media range. And next slide. So with this tiny amount of raw spingle, you already see a kind of red color. That's just the real image. And also it shows you the antibacterial results. And in the 30 minutes at this amount of this loaded raw spingle, you will see 4 log reduction, even reaching 6 log reduction, exposing to the light. More interestingly, it also will see no effect on the dark, but only on the slide. And also, this is another microorganism. This is the gram-positive deterrent, and also shows the good effect. Next slide. And also, we tested with a virus, but in this case, we use a plant virus. Is called the T7 bacteriophage. It's not a mammalian virus. It affects the bacteria, and also but it represents the viruses. And if the material could kill this type of virus, it would definitely kill. And I do have this evidence to show you in the next few slides. But here is, and this material would be able to you can hand wash 15 times and also expose continuously for light for 7 days, still possess the same type of the antivirus functions. And this is actually provided at the 30 minutes, you will see a very quick kill to the virus. So this is the fabric actually draw a lot of attention just in this week, in the last week, because there was a news release from American Chemical Society due to the publication. Next slide. So next one, I show you another reusable materials. This actually covers for the surgical face masks and also potentially for the N95. This goes with the microporous materials. We're going to use nanofibers. Also, we use light, and also will provide those functions outlined here. Next one. And also recently just published, there was also a lot of interest for this technology. So everyone is familiar with the surgical, the disposable face masks we see everywhere now. It's made of the 3 layers of materials. So now we call non-woven. Top and the bottom. The middle one actually is the filtering materials. And normally it's produced by the melt blown, non-woven. It's a micro-sized fibers and or sub-microstructures. But here we use electron spun membrane materials to replace this one. But on those materials, the fibers, we actually added those chemicals. Those are the vitamin Ks and obviously edible. And also there's 3 different derivatives. We all know that based on our computational modeling, this is the at molecular level modeling, we know they are photosensitive and also would be able to generate the reactive oxygen species under daylight. This is the visible, the white light. Next slide. So you also will see here is we generated the functions in producing these images and showing the antibacterial function and also antivirus function. But also here shows you the level of generated reactive oxygens. First shows a hydroxy radical. Second one shows hydrogen peroxide. Third one shows the singulated oxygen. And also the highest one actually come from the compound called VK3, vitamin K3. And very interesting because this is a slightly different mechanisms and through those photochemistry, but much more effective than the rose bingo. Next slide. So those materials also would be able to provide very quick kill. And also you see 30 minutes, this is the E. coli. It's a little bit longer time to kill this positive, the gram-positive bacteria. Also this face mask materials could be repeatedly used under the daylight. So it show you 5 times repeated use, you don't see any kind of reduction. And in fact, we recommend the users that you can use it until you feel it's physically soiled and you have to dispose it. So this is the antibacterial functions. It's also very, the next slide. And you're going to see the antivirus functions. So here also we use the T7 bacteriophage to demonstrate the virus activities. Also could be killed the virus and in 30 minutes a light exposure. And this actually is the one that we really use the mammalian virus and collaborating with the professor in the veterinary medicine. It used the cat coronavirus, same type of strains of our SARS-CoV. So 30 minutes, 3 log reduction on the surface of the materials. So this is the one for we call the single use or disposable face masks, or possibly for N95 respirators. Next slide. So that just shows you all these, the 2 different face masks we've been produced in our lab just in the past 6 months. And I have to acknowledge all this, the researchers. So first, the researchers collaboration from Professor Nitin's group for the microbiology test, and also Professor Brian Murphy and for the virus test. And also the doctor, patient Dr. Ahmad El-Marghazi, and also Mr. Zheng Zhang, and he's a graduate student. Those 3 contributed to the experiment. Also, we have to thank the funding agencies. Obviously, we did receive the the Kraft Fund from UC Davis for the COVID-19 related research. But in fact, those research current one will carry on research because this is for the food safety. We're using this photoactive agent for food and safety applications. Also, we're using another type of the positive charge for the sensor studies. So those were all the funding agencies. Thank you very much for your attention.
Thank you, Professor Sun. There's one question in the Q&A right now, and it's from Michael. For number one, is there any way to regenerate the surface chemistry, or would it be semi-disposable due to degradation?
Okay, so those chemicals are reusable, and also it's light refreshable, we call it. Okay, so light refreshable means that you have to expose to light. And so every time you use it and you can re-expose to the light, you're going to regenerate the function. Yeah. And also if you use it on the light, obviously would provide the function. And the WK3 type of the compounds, even more interesting, I did not show more details. And some of the structures would be able to retain the energy and obtain the under the lighting conditions and provide the function even under dark. So that's even more interesting for us. That's the one that we are continuing for the research for the N95s and respirator applications. So that means that it's not only just the on the light you have this function, on the light you can regenerate the function, and some of the chemicals will be able to retain the function when you are exposed to the light and refresh it as well. I don't know whether I answered the questions.
Thank you, Professor Sun. That was really interesting. Now, I'd like to turn it over to Professor Islam from the Electrical and Computer Engineering Department. Saif, are you there and ready?
I'm ready. Thank you very much. Good evening. It's my pleasure to share some of our research activities with all of you. Thanks for staying late for the last talk. So if you all remember, in early 2020, around July, around 200 scientists from all around the world signed a letter, sent it to World Health Organization and urged them to seriously consider the growing evidence that the coronavirus can be transmitted through the air. And right now, everyone believes that that's the fact. And like many other similar viruses, COVID-19 spreads through tiny droplets containing virus particles, and they're capable of causing infection. They can seriously go from one place to the other, remain suspended in the air for a long time. And if they're not in big droplets, they actually could move a long distance. So physical activities such as breathing, sneezing, or coughing of an infected person could increase the density of airborne viruses around them and could rapidly spread them around and could infect other people. So if you look at the video here, Francesca, if you could turn on the video. So here you are looking at on the top left of the video, there is a person who was breathing. And if you are normally breathing, you are spreading droplets for maybe a couple of inches to maybe a few feet. But if your breathing activity gets really strong, somehow the video stopped probably. If you continue to breathe, but engage in conversations, then you are spreading droplets far than a couple of inches or feet. It could be a couple of feet, and that's why this 6 feet requirement was imposed and introduced by the scientists. And these particles that we generate, some are very heavy. There are few microns in diameter. They would settle on the floor, on your clothes, on any surface. Others that are really small would fly far, far away. But if you sneeze or cough, or if you're in a protest or talking with lots of emotion and having heated discussions, then your droplets could go 20, 30 feet away and could remain suspended in air for a long time, for hours. In the next slide, I would show how an infected person in a room Actually, if you go to the, okay. So you probably can see an infected person in a room, and the biggest droplets in that room are all settling right in front of the person. But if you don't have ventilation in the room, if you can't really allow windows or air circulation, particle density in the room increases. So anyone taking care of the infected person is also in danger. Next slide, please. So we were trying to understand how, as an electrical engineer, how we can address some of these problems. The infected person in a room constantly creates viruses in the air on the surfaces. So you could use UV lamp to kill the viruses. The people who are taking care of the person, nurses and doctors, could have PPE and masks, and previous presentation was talking about some of the masks technologies people are developing. You can have good air circulation, you can have windows open, and HVAC system could take care of purifying the air. So we're trying to see, as electrical engineers, what else we could do. So one device that people use for purifying air, and many of you might have that at home, is air purifier. And that can be enhanced to now address these COVID-19 like problems. And we focused on making it smarter and capable of handling this kind of tiny particles. Next slide, please. So if you want to get rid of virus-like particles, let's see how air purifier would perform. So typically air purifier would use a HEPA filter, high efficiency particulate air filter, and they're good for particles of 1 micron or larger diameter. But if the particles are really tiny, they're not really as good. They're typically designed to filter out 0.3 micron or 300 nanometer particle sizes. But viruses are, the particular virus that are currently infecting us is around 120 nanometer or 0.12 micron. So that's smaller than the typical range of the HEPA filter. But in general, HEPA filter will do a good job in removing all the dust in your room, bacteria, and a lot of viruses too. But then it's not really designed to filter out this kind of tiny viruses. Next slide would show typically what HEPA filter's capability is. So here is a little plot on the y-axis. I'm showing the efficiency, and x-axis shows the size of the particles or viruses. So if you're 0.1 micron, which is the size of the virus, you effectively can remove 70 to 80% of the particles. If it is 0.3 or 0.2 micron, you actually don't even remove that much. You are in the range of 60%. So remaining 40% virus could still float around, could still go through the filter to the HVAC system, could go from one room to the other, and that's not really a good scenario. So one of the things engineers use in air purifier is a device called Ionizer, and next slide talks about ionization-based removal of particles. So imagine you have all these floating viruses in the room, and you attached one electron or one ion, positive ion, to the virus. And that way virus becomes an ionized virus. And that virus is a little easier to collect. It could settle on a floor and will stick there permanently. You can collect the ionized virus in a system with parallel plates of electrodes. And if it is negatively ionized, a positively charged electrode would collect it, the virus would glue to that surface, and you can kill it there by heating it and doing other things. Unfortunately, the way people trap this kind of ionized particles or generate those ionization particles typically uses high voltage, such as 10,000, 30,000, or 100,000 voltages. And when you bias something at such high voltage, you ionize the particles and dust and viruses, but you also generate an unwanted byproduct, which is ozone gas. Next slide, please. So if you are producing ozone while you're trying to ionize viruses, it's almost like you are spraying Clorox in your room and in your lung, and already your lung is weak because of COVID-19. With additional Clorox and ozone, you are actually causing more harm. So a purifier would remove virus, but then while removing, you are adding a poison to your lung, which might alternatively kill you, even if you are not killed by the viruses. So that's the problem. Ionizer is important because HEPA filter will not remove your viruses in any space. Ionizer would, but then ionizers are right now not approved by EPA or any agency because of their dangerous level of ozone in the in the machine that produces ions. So if you go to the next slide, so to summarize the problem, we need HEPA filter to capture viruses, but HEPA filter would not do a good job. So we need ionizers, but then ionizers would generate a lot of ozone because they operate at high voltage, and ozone is bad for lung. So as electrical engineers, we are trying to see whether we can produce ions, but not produce ozone in the room when we use this kind of machines. And to do that, you need to produce ions at low voltage. And that's where nanotechnology could play a role. If you go to the next slide, please. And if you can do this kind of ionization, that way you can use ionizers in buildings such as hospitals, schools, manufacturing plants, ships, submarines, hotels, or senior living facilities. And they could continuously circulate the air through such a machine and could filter out all the viruses and most of the viruses. And that way you can, if you don't have the vaccines yet, if your masks are not really doing the job 100%, you can use this additional capability to get rid of viruses from an enclosed space. Now, how do people ionize virus? They actually don't ionize virus. People attach a ionized particle such as electron or an ion. And typically when you remove an electron from an atom, it gets ionized. And you produce those ions, and those ions gets attached to the virus. And that way you can now do the next step, which is attaching the virus to something else oppositely ionized, and you capture it. Next slide, please. So this process typically is done by driving gas or air through two parallel plates. And you're looking at two parallel plates here. If you apply 10,000 to 100,000 voltages, whatever gas go through it would get ionized. So that's really good. And that ionized gas could then attach to the viruses to turn the viruses into ionized viruses. And the next job would be to then filter them out easily by using some techniques that people use in purifiers that we use at home. Next slide. So if you add a couple of spiky things such as nails or structures that have sharp tips, then you don't have to bias it at 100,000 voltage. You could reduce it considerably below 10,000 voltages because any sharp tip would have amplified field around it. So that's a trick people use, and that way you can reduce the voltage and still amplify the voltage around the peak to have a lot of ionized particle in a system. And if you go to the next slide, so the sharpest object scientists typically use these days for ionization is carbon nanotube. It's around 1 nanometer in diameter. And there is a little formula. I don't want you to really understand that, but if you look at the formula, it says field is voltage over radius of the sharp tip. So if your radius is small, your field gets amplified. And carbon nanotube, being the sharpest material in the planet, it is supposed to amplify the field the highest possible way, and that's what people use to ionize some gases. And here on the right, I'm showing number of different gases got ionized at 100 volt, 200 volt, 300 volt, and that's really good news. And we thought that way we can reduce the voltage quite a bit and reduce the production of ozone in an ionizer. If you go to the next slide. So what we also found is, in addition to using carbon nanotube like sharp material, which is actually not very stable when you are doing this kind of ionization process, the nanotube itself actually evaporates. It loses its existence after a little bit of time because the field is so strong at the tip, the carbon atoms simply evaporate. It's called desorption process. So we tested some semiconductor nanostructures, silicon nanowire, and made them super sharp, and found that when they're really sharp and you can do a little bit of engineering by adding impurities, basically by making them dirty. And we found that nitrogen, argon, or ammonia would ionize at 5 volt, 8 volt, or 9 volt. That's way lower than 100,000 voltage that you would use to ionize a gas. So when you are capable of ionizing something with your battery, you really are not producing ozone in your air purifier. And when ozone is not there, you basically would have the capability to produce ions without producing all these harmful ozone gases. So that's what we are currently doing. If you go to the next slide, Francesca. So here is little prototype that we developed with one parallel plate that you see on top. Another one is sitting underneath. There are a couple of spiky things, the silicon nanowires that helped us reduce the voltage quite a bit. And our plan is to implement it. If you go to the next slide, use this ionizer in a commercially available air purifier. We bought a number of them from different companies. We bought an ozone monitor, particle counter, and right now we are in the process of installing these ionizers that we produced in our lab in these machines. And then we'll test how the ozone concentration level is and eventually produce it to replace the commercially available filters, ionization filters, to produce ions and capture viruses in an enclosed space. Next slide, please. So the goal of this project is basically to produce ions without producing Clorox or ozone in the room. And if it works, it will help you in keeping your hospital, a room with infected people, safer for nurses and doctors and anyone around who are not infected with the virus. Next slide. So I have a little bit of physics in the next slide. I'll skip that through. And our project is still ongoing. Unfortunately, we still couldn't get our ozone meter because there is a huge supply shortage right now. The rest of the equipment and components are in our lab, and we are currently actively working on those. Thank you very much. Here is a little bit of physics that I'll skip unless there is a question. You can go to the next slide, which is the end. Thank you.
Thank you, Professor Islam. We do have a few questions before we wrap up. I think I will ask this correctly. Are you working on human safe to eyes and skin FAR UVC for killing viruses and bacterias 24/7 in public spaces?
So in an enclosed space, the concentration of virus is the highest. In public space outside a building, obviously the viruses are not really confined in a small space and you are not under too much of risk. But our goal is to keep cleaning the air in an enclosed room so that any infected people don't keep increasing the concentration of virus in a room. As we all know, virus now remains airborne for a long time. So with a device like this, you keep filtering the air. It's like a fish net. You keep catching all the fishes, as many as you can, and that way the concentration goes down with this device. HEPA filter would not do this because viruses are smaller than the capability of HEPA filter.
And last question, can ULPA filters be used rather than HEPA? We designed clean rooms with ULPA filters, which were chemically treated as a biocide.
Sure.
And this is from David Pegg.
Sure. I think multiple cascaded HEPA filter would also do a better job than a single layer of filter. ULPA filter can do this. Our goal is not only to use this kind of devices in a air purifier. We think eventually if this can be miniaturized, you could integrate it in your mask. You can make it really super tiny. A little battery can operate it, and that way it can be a portable device too. Other alternatives are not as portable as a technology that could be developed just by using ionization through a parallel plate and nanostructures in between them.
Fantastic. Thank you so much, Professor Islam. We are now out of time, and I apologize that we will not have enough time for an open Q&A session. Before we get to our raffle, I want to give a couple of additional shout outs to our alumni in Houston, Texas, La Jolla, California, and Denver, Colorado. I also saw that we actually have a UC Davis parent on tonight. So thank you for attending from so far away. And of course, many, many thanks to our Northern California alumni who continue to show up and support the College of Engineering at these events. Thank you for staying with us this whole time and attending. We encourage you to join us for these volunteer opportunity on December 17th. That's our Engineering 3 Intro to Design Fall Showcase. Help us evaluate our students' design projects. And in the spirit of Thanksgiving, thank you in advance for any and all that you do for the college. Our next Engineering on Tap will be a family-friendly event featuring Professor Ricardo Castro from Material Science and Engineering. He'll talk about how superhero technology could exist in the real world and run us through an experiment using chocolate bars. So bring the whole family for this fun event. It will be at 5 p.m. on January 21st. Another reminder to follow us on the College of Engineering social media pages, Instagram, Facebook, and LinkedIn. And Francesca, now to the raffle.